Cancer has marred many lives, bringing trauma and stealing loved ones, death and pain, scars for those who have overcome, yet the pain remains. As the second leading cause of death in the United States, according to CDC data from 2020, what is cancer and why does it harm so many lives? Simply put, cancer is uncontrolled cell proliferation. It happens when cells no longer respond to the mechanisms that control their growth. The uncontrolled cells can cause severe problems with various physiological functions as they multiply and steal nutrients and create excess waste as they try to sustain their rapid growth. Hopefully, by studying various methods that other animals, particularly the long-lived ones, use to suppress tumor growth, researchers may find other avenues for cancer prevention and treatment. Of particular interest for research are naked and blind mole rats and axolotls, as the rats, though ugly, are particularly long-lived, and the axolotls have incredible ability to heal and even regenerate their severed limbs. Naked mole rats are naturally inbred, which makes them a curious model for cancer research, as inbreeding typically leads to high rates of cancer and other genetic disorders. In a study, it was found that naked mole rat cells exhibit early contact inhibition, which means that the cells grow slower and halt at lower densities compared to other rodent cells. Early stopping could help slow down the spread of cancer. The cells were also found to be more resistant to becoming cancerous, as it required more mutations than usual to induce tumor growth. Blind mole rat cells also slow cell proliferation. However, they have their own way of doing it. They drink the proverbial Kool-Aid, and a generation of cells all commit suicide. In what is known as concerted cell death, the cells will multiply for several generations, and then suddenly, an entire generation will die, all at the same time. It was discovered that this unique generational cell suicide was a result of an interferon beta. This interferon was tested on both mouse and human cell lines and induced concerted cell death in the mouse line, yet not the human one. This is likely due to the difference in receptors for interferon beta in humans compared to the rodents. Axolotls, however, are quite different than rats, and they have been found to hold powerful mechanisms for reprogramming cancerous cells. Axolotls are excellent biological models for understanding the healing process and cancer resistance, as they are practical in the lab, plus they're cute. Cute salamanders that never grow up, and have the incredible ability to regenerate fully functional limbs. Now, to understand how impressive this is, when humans are injured, say it leaves a scar, the scar is merely connective tissue, holding everything together and providing a little bit of protection for the underlying tissue. But when an axolotl is injured, stem cells migrate to the area of the injury and produce the tissues needed for fully functional, whatever that limb or section was. Impressive, to say the least. Axolotl egg extracts were tested for their ability to reprogram cancerous cells, specifically breast cancer cells, and with positive results. They were even better at reprogramming the cancerous human cells than using human stem cells, and they are available at a fraction of the cost, saving both time and money. Plus, there's no ethics issues. In my personal opinion, it may be wise to study the axolotl genome, despite its daunting size, considering these results and their unique healing abilities. These promising results may lead to new cancer treatments. Understanding the various methods of cancer resistance by studying unique animal models can provide new information on the mechanisms of resistance and provide new avenues of treatment in the future. You never know. Maybe the ugly-looking animal you thought was useless will be the next cancer treatment. Thank you for your time. I hope you've enjoyed listening to new developments in cancer research.